Welcome to ImmerseCell. ImmerseCell is an immersive learning experience that allows you to explore the cell and its different organelle structures. Before we start with the learning experience, let's have a look at the controller in your hand. When you look at your controller, you will notice that the buttons are labeled with the actions they perform. In front of you, a button will appear. Go ahead and select the button by pointing at it with a laser and pressing the A button on the controller with your thumb. This is the way you will be selecting objects within the learning experience. Now, observe the mitochondrion in front of you. Practice grabbing the mitochondrion by pointing at it and pressing the button under your ring or middle finger. You can rotate the mitochondrion by moving the thumbstick left or right. Well done! Now move the mitochondrion forward or backward by moving your thumbstick forward or backward. Release the mitochondrion by pressing the button under your ring or middle finger. Great! Now that you can navigate with your controller, look to your right. You will see a screen highlighted in blue. Throughout the learning experience, this screen will display the information. In front of you, in the right corner of the room, is another screen highlighted in purple. This screen indicates the sections of the learning experience. The section in progress will be the one highlighted. After the introduction, you will be introduced to the mammalian cell. Then, you will explore cellular organelles, namely the nucleus, mitochondria, lysosomes, and autophagosomes. At the top of the screen, you can observe your progress throughout the learning experience. Two buttons will help you navigate through the learning experience. When the next button appears, you have to select it in order to move on to the next segment. The back button, when visible, will enable you to repeat what was previously said. Fantastic! It is now time for you to start interacting with the cellular structures. Remember that you are going to write a test upon completion of this lesson. Therefore, try to focus on the information as you observe the cellular structures. Alright, please select next if you are ready to proceed with. In front of you, you will see a three-dimensional artistic impression of the mammalian cell, just as it is typically depicted in a textbook. Go ahead, pick up the cell and move it around in order to observe its intricate structure. As you observe the cell, notice the various structures that the cell consists of. These structures are called organelles. In the learning experience, we will be exploring 5 different organelles. These organelles are the nucleus, mitochondria, lysosomes, and autophagosomes. Each organelle performs a specific function within the cell. The complex interplay of organelles determines a cell's function and contributes to its structural properties. The collective functional and structural characteristics of cells enables and define an organism's tissue structure and function. Please release the cell. The cell is the smallest structural and functional unit and living entity, serving as the building block for tissues, organs, organ systems, and the entire multicellular organism. Understanding each cellular structure is critical to really grasp the role of each structure in cell function and dysfunction. The cell is complex. It is three-dimensional, dynamic, and living. It responds to cues from its surrounding environment. This is challenging to depict in a standard student textbook. Therefore, this immersive learning experience is designed to expose you to the complexity of the cell in a different way. This immersive tool has been built using confocal microscopy data from real cells to make your learning experience much more tangible. This immersive tool has been built using confocal microscopy data from real cells to make your learning experience much more tangible. The pop-up screen displays three-dimensional images of selected organelles that have been acquired using confocal microscopy. Before we go on with this learning experience, it is important to know what information confocal microscopy images, otherwise known as confocal micrographs, contain. When acquiring a confocal micrograph of a cell specimen, each organelle is normally labeled with a fluorescent dye. This dye causes each organelle to emit light at a specific wavelength. This results in each organelle being represented with a specific color. This is challenging to depict in a standard student textbook. Therefore, this immersive learning experience is designed to expose you to the complexity of the cell in a different way. Pick up the screen and pull it towards and away from you in order to observe its structure. Take a moment and compare the artistic impression of the cell with the real cell. When you have completed exploring the microscopy images, please release the screen. Good work! You have now learned that numerous small functional structures called organelles combine to form a very complex, dynamic, and a living entity called the mammalian cell. Now it is time to learn about the main organelles in more detail, one at a time. Select Next if you are ready to proceed. The nucleus is typically the largest single organized cell component. It can be seen as a distinct spherical or oval structure, often located near the center of the cell. It is surrounded by a double-layered membrane, the nuclear envelope, which separates the nucleus from the cytoplasm. The nuclear envelope contains many nuclear pores, which allows the necessary traffic of molecules and proteins to move between the nucleus and the cytoplasm. The nucleus contains the genetic material of the cell known as DNA. DNA provides code that directs the production of specific structural proteins and proteins that serve as intracellular enzymes. DNA also controls the process of cell division, otherwise known as mitosis. It serves as a genetic blueprint that ensures that the cell produces another cell just like itself. By directing the amount and type of proteins being produced, the nucleus can be seen as indirectly governing cell activities, and therefore, the nucleus is typically seen as the control center of the cell. Go ahead and select the highlighted nucleus. On your right, you will see a three-dimensional model of the nucleus you have selected. This model was acquired using confocal microscopy. Go ahead and pick it up. Observe the structure of the nucleus by rotating it and pulling it towards you. DNA is packaged into regions of dense chromatin termed euchromatin. These regions are represented by the dark blue structures. DNA is also packaged in regions of less dense chromatin termed heterochromatin. These regions are represented by the light blue structures. Please release the confocal model. In this section, you have learned about the main organelle, the nucleus, which contains the genetic information of the cell and can be considered as controlling cellular activities. If you are ready to proceed with the learning experience, please select Next. In front of you, you will again see the artistic impression of the cell. Please select one of the mitochondria that are highlighted within the cell. Cells require a substantial amount of energy to remain functional and viable. Mitochondria generates energy in the form of adenosine triphosphate, also known as ATP. Mitochondria are often depicted as oval or rod-shaped structures. This can be seen by the mitochondrion on your left. Pick up the mitochondrion to observe it. Mitochondria are characterized by a double membrane, specifically a smooth outer membrane and an inner mitochondrial membrane. The inner mitochondrial membrane forms a series of enfoldings called cristae. The cristae contains proteins that make use of oxygen to convert the energy found in food into a usable form. You can see that the cristae project into an inner mitochondrial space filled with a gel-like solution known as the mitochondrial matrix. This matrix consists of hundreds of different dissolved enzymes. These enzymes prepare nutrient molecules for the final extraction of usable energy by the cristae proteins. Mitochondria contain their own DNA, distinct from the DNA house in the nucleus. Mitochondrial DNA encodes for many of the proteins required for the production of ATP. When you have completed your exploration of the mitochondrion, please release it. Remember that you can release the mitochondrion by pressing the button beneath your ring or middle finger. On your right, you will see a three-dimensional model of mitochondria situated around the nucleus. This model was acquired using confocal microscopy. You may pick up the confocal microscopy model of the mitochondria. Each individual red particle represents a single rod-shaped mitochondrion. In reality, it is not always the case that mitochondria are rod-shaped. You are able to see this by noticing that some mitochondria are fused together to form complex networks. Please release the confocal model. Displayed in front of you are three-dimensional time-lapse sequences of mitochondria that localize around the nucleus. When you look at the mitochondria in the time-lapse sequence, you will become aware that mitochondria are highly dynamic organelles that are in constant change. Note the segregation of mitochondria into smaller structures. This process is termed fission. Now notice the combination and joining of mitochondria into larger and longer structures. This process is termed fusion. Pick up one of the screens and pull it towards you. See if you can identify a few fission and fusion events. Fission and fusion are very important functional properties of the cell. Fission occurs to protect cell function by facilitating equal segregation of mitochondria during cell division. Fission is also a critical process to isolate segments of damaged or dysfunctional mitochondria. It promotes their clearance and removal through autophagosomes, which will be discussed later. Fusion, in turn, is an important mechanism required for proper respiratory activity. It ensures the metabolic efficiency of mitochondria. Well done! You have now learned that mitochondria produce energy for the cellular functions. You have also seen that mitochondria are highly dynamic organelles, segregating and joining to form smaller or longer structures through the processes of fission and fusion. Select Next when you are ready to explore the next organelle. Please select one of the highlighted lysosomes. On your left, you will see an artistic impression of the lysosome. Their main function is to digest intracellular material. Lysosomes are small spherical organelles. Instead of having a uniform structure, lysosomes vary in size and shape, depending on the size of the organelles they are digesting. Looking within the lysosome, one can see that its lumen contains a large number of different enzymes. These enzymes are known as hydrolases. Hydrolases allow the digestion of mainly proteins, but also carbohydrates, lipids, and nucleic acids. On your right, you will see numerous lysosomes in red, situated around the nucleus. Go ahead and grab the confocal model of the lysosomes on your right. Rotate and observe their structure. You will notice that the cell contains numerous lysosomes and that they vary a lot in size and shape. Displayed in front of you is a time-lapse sequence of lysosomes. You can see that the lysosomes are constantly moving. This movement is bidirectional and operates in a stop-and-go manner. Motor proteins, dynein and kinesin, that depend on ATP facilitate the transportation of lysosomes. This transportation happens along the tubulin strands, which you will be learning about in the next section. Great! In this section, you have learned that lysosomes serve as the intracellular digestive system and that they constantly move within the cell. Select Next when you are ready to explore the next organelle. In this section, you will be learning about autophagosomes. Many textbooks do not yet feature autophagosomes since their full discovery has been quite recent. You may proceed by selecting one of the autophagosomes. Autophagosomes are double membrane vesicles that are responsible for macroautophagy. Macroautophagy is the intracellular degradation process for damaged organelles and cytoplasmic cargo material. Macroautophagy is facilitated by autophagosomes and is a crucial process for the removal of toxic material and turnover of long-lived proteins. Let's look at how macroautophagy occurs. When the cell receives a stress signal, for example, depletion of nutrients, it produces a double membrane structure called the phagophore. The phagophore then sequesters damaged organelles and cytoplasmic cargo. The phagophore then expands around the cargo and forms a sealed vesicle called the autophagosome. Then a lysosome fuses with the autophagosome to form an autolysosome. Degradation takes place when the cargo is exposed to the lysosomal hydrolytic enzymes. During degradation, amino acids are produced and released back into the cell. There, They can be used for ATP generation or as building blocks to create new proteins. On your right, you will see a three-dimensional model of autophagosomes acquired using confocal microscopy. You may pick up the confocal model. Each individual green particle represents a single autophagosome. You can best observe their structure by rotating them. Please release the confocal model. In front of you, you will see time-lapse sequences displaying the trafficking of autophagosomes within the cell. Notice that they move towards and away from the cell center in a bidirectional manner, just like the lysosomes. As you can see, some are transported much faster than others. Please pick up one of the screens and take a moment to zoom in. Observe the trajectory of the autophagosomes in movement. You will notice that they do not move randomly, but along specific tracks, also mentioned in the lysosome and mitochondria sections. These tracks are formed by the tubulin network, an important part of the cytoskeleton. This network consists of tubulin strands composed of alpha and beta tubulin proteins. The cell assembles and disassembles this network dynamically as required. This is similar to new roads built in a city while old ones are removed. You can imagine that when this tubulin network breaks or collapses, there will be a major traffic jam. Mitochondria, lysosomes, and autophagosomes would start to accumulate. This causes the cell to become very dysfunctional and may lead to diseases.